Okay, I'm going to continue on with some more environmental factors that influence microorganisms. Microorganisms, so we've talked about temperature and oxygen requirements and how different species of microorganisms will differ in terms of their requirements for those two different things. And pH is no different, so different microorganisms have different pH requirements. Organisms that are neutrophiles are going to prefer pHs that are close to neutral, pH 7. If you're an acidophile, remember phile means to love, you like acidic pHs, so lower pHs. And if you're an alkalinophile, you like higher pHs. You enjoy or must grow in an alkaline type of environment. So neutrophiles, the optimal pH range, about 6.5 to 7.5. Hopefully you remember from your A&P classes that the human body, most human body fluids have a pH of 7.35 to 7.45. So that's within the neutrophile range. So most human pathogens are going to be neutrophiles. Acidophiles like acidic, lower pHs. And there are some extreme acidophiles that have been identified that even thrive near pH 0. So that's such a low pH, so acidic that if you stuck your hand into a pH 0 environment, the tissues would probably be destroyed almost immediately. But there are microorganisms that are have evolved to thrive in those types of environments. If you're an obligate acidophile, remember obligate means obligatory, you must live in an acidic pH. If you're an acid-tolerant microbe, though, that's kind of like facultative. Remember, facultative anaerobes can live in anaerobic environments, but they actually prefer an aerobic environment. An acidic-tolerant, an acid-tolerant microbe doesn't require an acidic environment, but can tolerate it. And a good example, Heliobacter pylori, we were talking about how that organism lives along the stomach lining. The stomach lining is a microaerophilic environment, if you remember that from the last lecture. And it's also an environment where there are acidic conditions along the inside lining of the stomach. So H. pylori doesn't need acidic conditions, but it can tolerate them, and so therefore it's one of the few things that can grow along the inside lining of the stomach. Alkalinophiles, there are alkalinophiles that have been discovered that thrive in conditions up to pH 11.5. All right, then osmotic pressure. Different microorganisms will vary according to the osmotic pressures they like. And remember, osmosis is governed by or controlled by solute concentration. Water is attracted to solutes. So now when we talk about a cell sitting in an environment that's isotonic, hypertonic, or hypotonic, that means relative to the intracellular fluids. So if you are an organism that's adapted to growing and thriving in very, very, very salty fluids, then a fluid that might be hypertonic for us would wind up being isotonic for them. So those terms are relative to your intracellular fluids and what you're adapted to. All right, so again, you want to go back and review what happens to a cell if it's sitting in an isotonic solution, a hypertonic solution, or a hypotonic solution. All right, so there are organisms that are called obligate halophiles. So halo here is a prefix referring to salt concentration. Salts like sodium are a type of solute. There are organisms that are adapted to thriving in water that contains a very, very high level of salts. And so those are called obligate halophiles if they must live in those environments. Now, can you figure out what would happen if you put an obligate halophile into fresh water? What would happen to it? So what you have to do is think about, okay, is that fresh water hypertonic or hypotonic or isotonic to these obligate halophiles? And hopefully you're thinking, okay, well, these organisms live in salty water, so salty water would be isotonic to them. If you put them in fresh water, solute concentrations are going to be too low, so that's going to be a hypotonic environment. And when a cell's in a hypotonic environment, remember the water's going to flow towards where you have more solutes. So there are going to be more solutes inside the cell. That's going to make that cell swell. Facultative halophiles. So that means they don't like necessarily salty conditions, but they can tolerate them. A good example, Staphylococcus aureus. I mentioned that organism will come up a lot this semester. That's an organism that can live on your skin. It lives in the nasal cavities of a lot of people, and those are all environments that are pretty high in salt concentrations, sodium concentrations. And it doesn't need those environments to survive, but it can tolerate them, whereas a lot of other things cannot. So it's considered to be a facultative halophile. So this is osmotic pressure. Your hydrostatic pressure can also influence your growth. So there are microorganisms, for example, that live way down deep in the ocean, miles under the surface of the ocean. And there you have very, very high hydrostatic pressures. And pressures that would crush a lot of organisms that naturally live up here on the surface of the earth or up higher in the water. So those organisms have to be adapted to very high hydrostatic types of pressure. Radiation tolerance is another physical factor that can influence growth. And I mentioned in the first clip for this chapter that radiation includes any type of energy that travels in a ray or a wave. And so that includes visible light, microwaves, UV light, ultraviolet light from the sun, gamma rays. Those are really high energy rays that we associate with dangerous types of radiation. All right, so believe it or not, good old visible light is actually toxic to many types of bacteria, because when those visible light rays hit molecules inside the cells, they can actually be damaged from that. And you guys will learn, if you ever take a chemistry class, you'll learn more about what visible light can do as triggering chemical reactions. Remember all those reactive oxygen molecules we talked about in the last video lecture? Visible light can trigger, when it's one of the things that will trigger the formation of those. And if you accumulate enough of those without inactivating them, getting rid of them, they can wind up damaging your DNA and your other important molecules, and you wind up dying. All right, so some types of bacteria make pigments. They're called carotenoid pigments. Kind of sounds like carrot. And these tend to have yellow and orangish or goldish types of colors. And what those pigments do, the reason they have color, is because they're absorbing most visible light. They're reflecting back only certain wavelengths of visible light, and those are the ones that you can actually detect as particular colors with the eye. So those carotenoid pigments soak up a lot of the visible light rays and help protect the bacteria from being damaged by those. And here's Staphylococcus aureus. Again, aureus actually refers to gold. This organism has a goldish, yellowish, or even orangish type of color when you grow it on an agar culture medium in the laboratory. That color is coming from these carotenoid pigments. So you would see in a laboratory setting that incubators where you grow bacteria, usually they don't have clear windows on them. They either have no windows or they have smoky windows that block out a lot of the visible light. More powerful types of radiation, like ultraviolet light and gamma radiation, are actually used deliberately to kill microbes when needed. And interestingly, hospitals, there are some studies that are ongoing these days. There's a link here to a video you might want to check out, where hospitals are actually just beginning to use visible light to help control the number of microorganisms in patient rooms and operating rooms. So basically you flood the room with intense visible light, and a lot of microorganisms can't handle that, and it winds up killing them. So that's pretty interesting, the idea that you could use visible light, intense visible light, to actually kill a lot of microorganisms. And that's much safer for the workers in the hospital. They don't have to get exposed to ultraviolet light or something more intense as a type of radiation to help control microorganisms. All right, so I'm going to cut that video lecture off here. Thankfully not as long as the last one. The next video clip will continue on with some additional environmental factors.